Okay, DNA fingerprinting, as the name implies, is a unique look to DNA, in this case the fragments. As you can see here on the screen, it looks like, of course, I touched the screen there and it's big fingerprint here, but we'll clean that up a little bit later. Looking here at deciphering out DNA and how it can be unique to particular individuals and what that can tell us. Alright, so our fingerprinting, each person's DNA pattern different? It's a good question to ask. We have actually sections of what's commonly referred to as junk DNA that doesn't really encode for proteins. It's made of repeated patterns. Each person may have different number of repeats, and many sites in our 23 chromosomes have different repeated patterns. These different patterns, just like the patterns on our fingers, give us what we call the DNA fingerprint, identify us as unique. And this is just an example here of looking at all the different G's and C's combos, the different patterns that do occur, and how different individuals, if these two individuals have different repeated patterns here. I highlighted those in green so you can hopefully stand out a little bit better. So DNA patterns for DNA fingerprints. We have allele 1, and I've indicated a couple of things here. Our cut sites. Remember, our restriction enzymes are cutting at very specific sites, and these in red are the same cut sites. The same restriction enzyme will cut at both of those sites. So we cut the DNA. You notice here, try to separate out, cut the DNA there a little bit. Here's our section 1, our section 2, and our section 3. How this would look when we run a gel. Section 1, section 2, and section 3. Try to put the yellow one here and our two other darker regions here for allele 1. Now, difference between two people. Now we have person 2 with more repeats. Okay, we go through, we cut that section out. Here's our section 1, our section 2, and our section 3. And as we can see here, we're getting a different run of the gel. These two cut sites, 1 and 3, are the same size, but this section 2 is different. There's more junk DNA essentially here, causing it to be a different run on the gel. So our DNA fingerprints are comparing our blood samples of defense clothing on that of a victim. You can see here's mom, here's dad, and then we're looking at other potential matches here on the offspring. And there are going to be differences in offspring comparing, because they're through recombinant DNA, through crossing over, through independent assortment and all that, you're going to get slightly different DNA sequences, but you're still going to notice, odds are, some similarities. And the example here, comparing the defendant's blood, the victim's blood, and then potential blood from defendant's clothes, we're able, again, to develop this fingerprint to get sort of a comparison here. And we could see where victim's blood may have gotten cross-contaminated here. This is some of the power of genetics that's used. So this process is what used to determine if two samples are from the same source. We're looking at DNA as two sources of fragments with restriction enzymes. Remember, those restriction enzymes cut at very specific regions. Fragments are separated using the gel electrophoresis and transferred to the filler, and then here we're able to probe and compare them. Also used for evolutionary relationships. Compare DNA samples from different organisms to measure how closely they may be related genetically. So we have our DNA for sample 2, sample 3, sample 4, and sample 5. Remember, the larger segments are up here, the smaller ones are down here. And this allows us to see how a turtle, a snake, a rat, a squirrel, and a fruit fly all may interrelate, on what commonalities they may have, what may be similar, and what might be more distantly related. So medical diagnosis, comparing normal alleles to a disease allele. Here's a chromosome with a normal allele and a disease-causing allele. Again, we run those alleles on the gel, and we see that they do cut at different regions. An example would be one of Huntington's disease. We're looking to identify those different alleles. And again, you say, well, they're the same length, right? How can there be different sizes to DNA fragments if they're the same length? While they are in total the same length, there's different sequences, and those restriction enzymes are going to cut this at different portions, giving it to different fragments than this allele here. forensics. This is where we commonly associate things with comparing samples from a crime scene of a suspect and a victim. Here's our suspect, here's our crime scene sample, I should say. Volunteer here, or our victim, and we're looking at 3 different suspects. So the victim typically will volunteer with their sample. We have the crime scene sample, and now we need to determine potentially which one of these 3 subjects, if there's any correlation or matching that occurs. Suspect 1, Suspect 2, Suspect 3, comparing to the crime scene sample, so we're looking for commonalities. So initially we look at this one and we say, well, Suspect 2 and 3 both share this. However, we need to compare all of the bands to verify the true fingerprint. And we see as we get down here, Suspect 2 matches not only this band, but also this band, this band, and this band, indicating Suspect 2 is a very close match for the sample at the crime scene that is different than the victim. And our Suspect 2 is the most likely match here. So RFLP, restriction fragment length polymorphism, and electrophoresis is used in forensic science. So we could be getting a semen sample from a rapist and a blood sample from the suspect, running it against the standard, and allowing us to develop a sense of comparison of what may match up. Because DNA fingerprintings are unique, this can allow us to eliminate other potential suspects and get close to identifying an individual, or determine whether an individual of interest has left DNA behind and has a potential to be there on the scene and contribute something in some way. So the RFLP, again, these are just markers that are used, and we're looking at different cut sites. Remember, those restriction enzymes are going to read right along and cut at very specific regions. As we see here, there are more cut sites indicating smaller fragments. Here there's only one cut site, a very large fragment, not running very far in the gel. So we can compare this to a real-world example here. Evidence of a murder trial. Do you think the suspect is guilty? So here's our standard, and here's what we're comparing. So blood sample 1 from the crime scene, blood sample 2, blood sample 3. And try to indicate here. Again, I did get this PowerPoint online. I'll look in the description for the links where I cite the sources here. I added some pictures, which are also indicated as far as where I got those. So we're looking at, it's a little faint, might be a little hard to initially see, but if we look at these regions right here, we can clearly see there's some bands occurring. Try to mark those with some arrows there. Blood sample 2 from the crime scene, notice there's a greater collection here, and blood sample 3. It's kind of those 3 matching, this matching, and this matching. When we compare blood sample 1 and 3, we see a lot of commonalities. Blood sample 2, not as many. So, blood sample from the victim. A little hard to see again in this image, but very closely matching the blood sample number 2 listed above. Blood sample from the victim here. We notice a little bit of a different pattern. And blood sample from the suspect. We could see, again, the circles and the colors, hopefully try to match it up a little bit closer. And the blood sample here was from O.J. Simpson. The victim here was Nicole Brown. And then Ron Goldman. So myself up here a little bit. And we could see where our suspects and victims and crime scene, how we're starting now to develop a pattern of what we're kind of seeing, what matches up with what. This is some of the power that a DNA kind of can serve to try to help us correlate if someone was potentially there because of the unique fingerprints that individuals have in their DNA. And then Nicole Brown here. So how gel looks in real life, again, that, as you saw in that example, they may not always be perfectly clear. There's typically a DNA ladder loaded, and that tells us what the size the bands are. And that allows us to compare. These thicker bands here indicating there's more DNA of that particular size present in the sample. And you can see here they're not always super easy to read. So for paternity test, here's mom's DNA, here's the child's DNA, and we have 2 potential father figures. We're looking at determining which father, if any of these, match up. So there's father 1, there's father 2, compared to the child. We notice the child shares these bands and these commonalities. Getting father 2 in this case is the most likely father of the child here whose DNA has been sequenced. So hopefully this was helpful. There's some other great videos on YouTube. You can also go through, and if you click on these links, again, the links to these are provided in the description. Go to the slideshow. You can actually try the process of PCR yourself virtually, and also electrophoresis. I have videos posted on both of these that you're welcome to look at to hopefully supply you with some good information to develop a good understanding.